So thank you for the introduction, and thank you all for participating today. I will talk a little bit about what we are trying to do here at Purdue in terms of how it relates to protein engineering and production. So as Jill was mentioning, I'm the director of the Molecular Evolution Protein Engineering Production Facility. We are located in the Discovery Park at Purdue University in the Discovery Learning Research Center building, and we are part of the Purdue Institute for Inflammation, Immunology, and Infectious Diseases. I know there are a lot of acronyms, but don't worry about it. I provided also a link to the facility at the down of the page so that you can look it up if you need to later. We, at the moment, are a discovery resource facility. That means that we are running collaborative projects with different PIs from Purdue and other universities as well. And we have various collaboration on research projects with industry partners. In general, I wanted to make an overview of how we are organized and what topics are we actually addressing. One part of it is the protein discovery part, and the other part is the protein production and manufacturing. In terms of protein discovery, we are focusing a lot on molecular evolution and protein engineering, because for a lot of applications, we cannot just use the enzymes or proteins that are available in nature, and we need to modify them so that they fit our needs. In terms of how we produce the mutant libraries, we are starting from either random approaches that just by using error-prone PCR, mutated strains, or recombination methods that produce mutated libraries of 10 to the power of 10 to 10 to the power of 12 different clones. We also use semi-rational approaches in which we are looking at the diversity present in the natural evolution and try to pinpoint what are the parts of the proteins that were conserved during natural evolution and try to minimize the areas of the proteins that need to be mutated. This decreases the library size significantly, and we only need to look at 10 to the power of 4 to 10 to the power of 5 different clones. And of course, the most important part would be if we would have a rational approach in which we could say, okay, this mutation would have this effect on the protein and it will give us the property that we are looking for. However, that is still not very well understood in the field, but there are more and more computational methods and tools that are becoming available, a lot of them now based on machine learning and artificial intelligence. And of course, this creating libraries like this would minimize the amount of clones that need to be screened for the property of interest. We are also looking sometimes at the natural diversity because nature already did in some cases the job for us during millions of years of evolution. And to do that, we are either isolating the genes or taking directly the cells that are expressing the protein of interest, in some cases directly or by applying some methods of quorum sensing disruption so that we can isolate more microorganisms and then go into the functional screening itself. So a lot of these methods are presented more having in mind the discovery of new enzymes from microbial strains, in which cases people start from microbiome and diversity that they find in different environments, like even just soil or salt water, depending on what you're looking for. In some cases, if we are looking for thermostable enzyme, we would probably start with microorganisms that are growing in hot springs. If we are looking for wood-degrading enzyme, we would probably start from the microbiome of the gut from the insects. Either cases, a lot of people are focusing on metagenome and microbiome sequencing-based approaches. We don't do that so much. We are mostly developing functional screening, and for that we either take the diversity, isolate the gene, and try to recombinantly express them in different strains, or we try to isolate the microbes that are producing the function of interest, but first we need to disrupt the quorum sensing because microbial communities secrete all sorts of signal that prevent the growth of other microbes from that environment. It is actually estimated that less than 1% of the microbes from any environment can be cultured in laboratory conditions, and by using quorum sensing disruption methods, we can actually increase that number. As I said, this slide is originally intended with having in mind the discovery of new enzymes from different environments, but in the end, we are applying the same procedure where we are screening for new antibodies, either from directly animals that have been immunized, and we are looking for B cells with specific antibody that bind the antigens that we are interested in, or screening hybridoma cell lines that we have produced, or B cells directly from human patients. In the end, it does boil down to the functional screening, and it is very important that we know what we screen for. And as you can imagine, that we are talking about enzyme, and each enzyme has a different activity. Therefore, we need to optimize the screening for each individual protein that we are planning to screen for. And also for each function that we are looking for, whether we are looking for thermostability, increased thermostability, increased pH stability, binding to a specific antigens, if we are talking about antibodies and so on. So the screening system is one of the things that we are highly focusing on. And as I said, it's very project specific and property specific. The correct assay that reflects the property of interest is very important, and this basically consumes most of our times when we start a new project. And in terms of screening method, we are basing our screening either on microtiter plates or flow cytometry and microfluidic devices. Like you can see in the videos below, this is an example of microfluidic devices which can highly speed up the screening process, because we can have and look at the properties of each individual cells in individual compartments. Once we have the protein of interest, we basically move on to cell line development and see how we can produce that protein. We can take it in our facility to a certain yield and a certain scale up, but in the end we need to be compatible with the industry that will take it further for actual production. So the cell lines platform that we are working with currently are either bacterial, and we are working with E. coli and Bacillus species. We have yeast expression system based on Pichia and Saccharomyces. We have animal cells, CHO and HEX cell lines. And also we are working with in vitro transcription translation system, so far only from E. coli, but we are planning to expand it to plant insect cells and yeast cells. By having this production platform and expression cell lines, we can basically cover a wide range of proteins, starting from antibody, vaccines, cytokines, and enzymes. Once we have the protein of interest that we want to express, we need to also spend quite a lot of time in developing a stable cell line for expression. Even if we are talking about E. coli or yeast, where we can have a selection system that is based on antibiotics, this can also become really expensive on the long term if we are talking about hundreds of liters of fermentation. So in general, we are trying to do stable integration into the cell line genome. This is even more important when we are talking about mammalian cell line, because transient expression costs are normally ranging around $2,000 per liter. So there is no way that you can do 100-liter fermentation on a transient expression platform. We are also doing a lot of condition optimization of the fermentation based on a design of experiment approach. Again, it is very important to stabilize the conditions or the optimal conditions for highest production level, because if we are scaling it up, we need to have optimal performance, and we cannot waste the resources in the final scale-up process, because it all adds up to the cost of the final product. We are equipped to do small-scale production in the lab. Of course, we have a lot of shakers and incubators for flask fermentations, but we are also equipped with real fermenters that can do 20-liter fermentation for the microbial cell lines and up to 40-liter fermentation for the mammalian cell lines. Our fermenters are thought in a way and are bought from a company, Sartorius, that is one of the most commonly used in industrial environment, and their fermenters are offering ease of scale up, that the conditions that are set up in this small fermentation scale can be easily adapted for large production later on. This is, I would say, low to medium production level, even though it's a lot more than a lot of academic partners have available. So we can still do quite a lot of protein in this system that can even go sometimes in preclinical trial or clinical trial. We are not set up for doing GMP level, so we cannot do clinical trial on human, but we can have some animal, and we are working on a collaboration with the veterinary school where they are doing some antibody testing in clinical trials in dogs. The next step would be protein purification. So the downstream processing, once we have the fermentation, we basically separate the cells from the media. And depending on where our protein is, we can take it to further step, either by concentrating, by a series of filter, ultrafiltration system. We can change the buffers at high speed and high volumes that we are working with. We also have a cell homogenizer, a French press, that we can actually disrupt the cells in case our proteins are intracellular. Then we take them down to the chromatography step for multiple purifications. We have a couple of high throughput systems that can also do high volumes of chromatography purification. Our ACTA system can do up to 150 milliliters per minute. So it can work also with really big columns. After we have, of course, quality control and assurance and the different assays that we can test at the end of the fermentation process. Mostly we are focusing on biochemical analysis, determining the kinetic constants for enzymes and antibody, and we do a lot of binding studies based on surface plasma resonance or ELISA. And we have in the lab automated ELISA washer systems that can do 50 plates independently without the need of a user, and the same for ELISA reader or for measuring enzyme kinetics. And we have two surface plasma resonance systems that are available in the lab. One, the BioCore X100, that can do up to 12 samples at the same time. And the BioCore 8K that can actually do 4 plates of 384 samples. So it's really high throughput. And now this is kind of an overview of what we have in the lab and the steps that we are doing for the whole platform of protein discovery and production. And now I will try to give a more detailed example of one of the proteins that we were using for some application, and more specifically the direct evolution of glucose oxidase. We are working at the moment with multiple enzymes, but they are in collaboration with industrial partners, and therefore I would not go into details about those projects. The direct evolution of glucose oxidase was a project that I was doing not at Purdue, but at my previous work, but it has already been published. However, the methods are exactly the same as what we are doing in the current project with industry. So I'm going to use this as an example for giving you a better understanding of how we work and what we actually do during one of these research projects. So why did we start with glucose oxidase? Well, we had at that time also another collaboration partner that was interested in multiple applications, but glucose oxidase in general is very important for industry because it is used in chemical industry for gluconic acid production. It has application in food industry for removing oxygen or glucose from beverages. It is actually used in the bread industry. So a lot of flours actually have glucose oxidase inside or other oxidases. But maybe one of the most known application is in pharmaceutical industry and medical field, where glucose oxidase is used on the strips that diabetics are using for determining their blood glucose. And why direct evolution? Well, because nature sometimes made the enzymes to work in the specific conditions that are needed for the organism's survival, not for our industrial application. So in some cases, like in the case of glucose oxidase, we do need different pH stability, and all enzymes, if they have increased temperature stability, are definitely stable for much longer, and they have a longer shelf life. In some cases, there is also a need for a different substrate specificity compared to the natural enzyme that you found in nature. So in all these cases, you can use directed evolution to change the property of the wild type version of the enzymes that you found in nature so that you can fit it to the application that is needed. Also, for industrial point of view, increasing the enzyme activity, even if it is for the natural substrate, is very important because most often the biocatalyst is actually the limiting step or the driving cost for the final product. Therefore, even if we increase the activity by 2 times, this means that we can decrease the cost by 2 times of the final product. So what is actually direct evolution? Well, we start from the single gene that normally encodes the wild type version of the enzyme that we are interested in, or the protein if we are talking about antibodies. We are introducing mutations at the gene level, so we are creating a pool of genes, each with their own individual mutation. We need to clone in specific vectors and express them in appropriate strains. In most of the cases, because of the transformation efficiency, as you can remember that some of the libraries can be quite large, we need to use either E. coli or yeast cells because they have high enough transformation efficiency to actually create a big enough pool of mutant libraries, of mutant variants. And the next step is the functional screening, in which we apply a method to select for the property of interest. We pre-select some of the mutants and we introduce new round of mutations and go on until we find the property at the level that we are interested in. As I was mentioning during this talk before, finding the right screening assay that reflects the property of interest is always very, very important because you always get what you screen for. So if you have the wrong assay or the artificial assay, you might get a variant that works on the wrong product, which is useless for the final application. In the case of glucose oxidase, which catalyzes the reaction of glucose to gluconic acid and reduction of oxygen to hydrogen peroxide, since we were interested in increasing the activity of the enzyme, we decided that the best option is to develop an assay that is based on the detection of the product of the enzymatic reaction, in this case being hydrogen peroxide. And we did this by coupling the reaction with a horseradish peroxidase and by adding an aminophenolxycarboxycoumarin substrate, which in the presence of peroxide and HRP, we have of horseradish peroxidase, we have the production of a fluorescent product that can be detected in microtiter plate or using flow cytometry or the microfluidic devices, as I will later point out. As I mentioned, the numbers can be really high and we can produce 10 to the power of 12 different clones or different genes by just doing error-prone PCR methods. But this diversity is lost because we have low transformation efficiency in yeast or E. coli cells. In the case of glucose oxidase, there the expression in E. coli does not work because of the folding, and it's only expressed in inclusion body. Therefore, it's not a functional enzyme. So we needed to use yeast cell for the production and for the screening in the library step. But as you can see, most of the diversity is lost because all the screening methods that are available cannot process typically more than 10 to the power of 5 colonies. But still, most of groups and all directed evolution approaches are highly based on microtiter plate methods. This gives a medium throughput, and of course, it's very expensive, especially if we are talking about the need of use of fluorescent substrates, which even if we are using 100 microliter of volume in each well of the plate, we are still talking about, in the case of 10 to the power of 5, we need to screen 1,000 plates. And of course, it is very time consuming. So these 1,000 plates could be screened maybe in a couple of months by labs that have highly automated robots. It is not really easy to do in an academic environment. Therefore, we were focusing a lot on developing ultra high throughput screening methods that were based either on flow cytometry or microfluidic devices. These actually are very low operating costs. Of course, there is a cost associated with acquiring the instruments needed for this type of screen. But once you have the instruments, the actual screening of the whole library is very low compared to the microtiter plate system, because we are using the same amount of substrate that is needed here in one well of the plate for screening the entire library. This is possible because we are doing single cell screening at a time. However, the other disadvantages are that the detection mode is limited to fluorescence. Therefore, we need to develop an assay that leads to the formation of the fluorescent product, and that assay needs to be reflective of the property of interest. And also the connection between the gene and the product must be kept during the entire time of the screening. In the case of the microtiter plate, each well contains one clone that produces one version of the enzyme or of the protein. In the case of the flow cytometry or microfluidic devices, we need other methods to keep that compartmentalization and connection. And the way that we are doing this is by doing droplets. I don't know why this video is not, okay. So what we are doing is introducing cells together with the substrate inside individual compartments. So each cell is responsible for expressing one version of the mutant enzyme. We are doing the reaction inside the droplets, so we lead the formation of a fluorescent product, after which we can actually put it on another chip and do the sorting for the droplets that actually show the property and the activity that we are interested in. We did this for glucose oxidase, and originally we started with completely random library where we produce a library size of 10 to the power of 7. After we got some preliminary information based on the original screen, we continued with a semi-rational approach where we narrowed down the parts of the enzymes that we wanted to further mutate. And in later steps, we went with a rational design where we looked at specific amino acids on the protein structure to actually mutate them and to increase the oxidative stability. So of course, in each type of library, we decrease the number of mutants that we need to screen, and we actually get higher quality mutants and better properties that we are interested in. So one of this screen actually gave us 5 mutants with at least 5 times improved in activity for glucose. We actually did some crystallization studies and molecular dynamic simulation, and we proved that even though the mutations were on the surface and away from the active site, the actual active site of the enzyme was modified. And it was more positioned in the right transitional state to catalyze the reaction. This brings me back to my point that it's very difficult to really predict how the mutations are going to have an effect on the final activity, because in this case, the mutations are nowhere close to the active site, but they still show that they can influence the activity in itself. We went one step further. So in the case of the using of the glucose oxidase for the biosensor application, we actually wanted the electrons go from the glucose all the way to the mediator and give an electrical signal. And we developed a new assay that can actually mimic the transfer of electron to a mediator, and we can detect this also using flow cytometry or microfluidic devices. And at the same time, with the same library, we could detect, on the other hand, the activity for oxygen. So we can have two competitive reactions, and we can select the clones that have higher activity for the mediator versus for the oxygen in this library. The most common mediators that are used in industry are ferrocene-based or nitrozoanilin-based. And we selected from this library, from the original screen using flow cytometry, we selected like 50 mutants, and then we did full characterization of the kinetic constants, both for for glucose and oxygen reaction, for glucose and ferrocene methanol, and for glucose and nitrozoanilin reaction. And we are collaborating with a company that was doing a structure-activity relationship based on machine learning approaches. We inputted the data that we got from our screens, and they predicted new mutations, a new combination of mutations that can further increase the activity for the desired mediators. And we found a mutant that has 12 times increased specificity for ferrocene, for example, and another one that has 8 times increase for nitrozoanilin. In terms of the oxidative stability, and this is for the library that we did a completely rational approach, we have noticed, so the problem with the enzyme is that once it is on the electrode, it's very easily oxidized and it doesn't last for long. And in some of the application where you would want a continuous glucose monitoring, for example, the enzyme gets inactivated very fast. And we noticed that the problem is also because the methionines on the close to the active site of the enzyme are getting oxidized, and this leads to a loss in the activity. So we did a very targeted approach where we did saturation mutagenesis at all the methionines on the glucose oxidase, and we found some combination of mutations that lead to an increase of 4 times in the oxidative stability. This is in short what we were doing for the glucose oxidase approach, but we did similar approaches and developed screening systems based on flow cytometry and microfluidic devices for other enzyme classes like cellulases, which also have a lot of industrial applications. Most commonly maybe used in biofuel industry and in laundry detergent. We also worked on peroxidases, which are used in lignocellulose degradation, again for biofuel application. They can be used in degradation of pesticides. They are used in a lot of water purification system and also in diagnostic kits. And again, we apply the same methods for xylanases and chitinases. Again, most application in pulp and paper industry and the biofuel cell industry. Now I will switch a bit gears, although it might sound like a completely different topic. It's actually not. We also applied similar systems for working on allergen immunization. There are a lot of people, apparently more than 25% of the population, that have some type of IgE-mediated allergy. And we were working on allergen-specific immunotherapy so that we can try to basically immunize and desensitize the people to the specific allergen. One of the projects that I was working on collaboration with the university from Serbia, They were interested in kiwi fruit sensitization, and apparently I was not aware that there are so many people that are having allergic reaction to kiwi fruit as well, but apparently 1.8% of the population shows sensitivity to this. Anyway, our approach was we actually did a library of allergens. So we expressed on the surface of the yeast cell, so displayed it on the surface, a library of allergens. So we introduced random mutations in one of the specific proteins that is expressed in kiwi and apparently is responsible for creating the allergic reaction. We created a library and then we screened that library against patients, so people that actually had allergy to kiwi. We took their serum and we tried to see which mutant of this allergen actually gives a good response on the antibodies for IgG versus IgE. So IgE are the antibodies that are creating or that are releasing an allergic reaction, and we don't want them to be released. So if IgG antibodies are present, they are basically neutralizing the antigen and preventing the immune reaction. So we did this also using flow cytometry approaches, and we selected some allergens that give a good IgG response and non-IgE response. And they were actually testing them in real patients, and they showed some improvements in terms of allergic reaction to kiwi. I mean, the same approach can be applied for multiple allergies and systems. So in the end, what we were mostly interested in was the screening system and the library development. Recently, we started a collaboration project with the veterinary school here at Purdue, and the Humane Society is interested in developing anti-pregnancy vaccine for control of wild horse population. Apparently the wild horses in the US are actually no longer really free range. They are confined in specific areas, in reservation. And since they do not have anymore enough of their natural predators, they could expand if they are not controlled, and they pose an ecological problem. There are vaccines that are currently being used. Those are based on using porcine ovary proteins, where basically from slaughterhouses, they take the ovaries from pigs, they just homogenize them, mix them with different adjuvants, and try to inject horses. However, these vaccines are very poorly characterized. They are not very efficient, probably because they are not very clearly defined. It's pretty complicated, or it's labor intensive to prepare them. There is no batch to batch consistency. It's also apparently quite difficult to obtain these ovaries from the slaughterhouses. And of course, by using it from other animals, you can potentially add contamination with infectious agents in the wild horse population, and you would want to avoid that. So that's why we started working on recombinant vaccine production, and the plan is to look at different proteins that are in the zona pellucida of the egg. Most common or prevalent proteins are zona pellucida protein 2 and 3. There are 4 in total, but for now we will focus on those 2. The idea is that if we use this protein in a vaccine formulation and inject the horses, they will produce antibodies against these proteins, and those antibodies would block the surface of the egg and prevent the sperm from reaching and producing a pregnancy. So this project just started. We are now looking at different parts of the proteins that we would want to express. There have been publications for quite some time that they are trying to express these proteins, and they prove to be relatively difficult. The reason for that is that these proteins are normally polymerizing in the zona pellucida area, so they are made to interact with each other, which makes it very complicated for them to be produced and in a soluble form, which is what we actually need for the vaccine. So we have a couple of ideas of how to actually use some hydrophobic patches that can block the hydrophobic part of the proteins, and of course remove the transmembrane domain, so that we actually have the proteins produced in soluble form. Another project that we are also working with in collaboration with veterinary school is developing PD-1 and PD-L1 antibodies for immunotherapy in dogs. Apparently, the tumor cells are expressing or upregulating the expression of PD-L1 receptor on the surface, and this somehow inhibits and triggers apoptosis of the tumor infiltrating cells that should actually kill those tumor cells. There are already a couple of drugs that have been approved by the FDA for human treatment that are specifically anti-PD-L1 antibodies. And basically, we are just using the same approach for developing similar antibodies and therapies for dogs. So in collaboration with the Purdue Institute for Drug Discovery and the veterinary school, they have did the immunization of the mice to produce hybridoma cell lines, selected the antibodies that are binding for the PD-L1 molecule, and then clone them so that we can recombinantly express. And this is what we are doing at the moment. We are producing the anti-PD-L1 antibodies in higher amounts so that they can use them in drug trials for dogs. And I think there were already a couple of dogs that were included in the trial so far. That's kind of a general overview of what we are doing. We are mostly working in collaborations at the moment, either with academia or industrial partners. The way we structure things is either we apply for joint project with PIs from academia, or we actually get a research project directly from industry. And we are focusing on the antibody protein expression and purification, on the development of new enzymes and protein, optimization of the functions for different applications. We are developing a lot of new screening technology, high throughput based on microfluidic systems, and development of new assay, because as I was mentioning, every enzyme and every protein is a specific case. We are doing, although we didn't focus on that so much, the strain and cell line engineering. This is really important once we have a finalized protein that we want to scale up for full production. And we are doing also a lot of binding and kinetic studies, both for antibody and for proteins, for enzymes. So thank you for your attention, and if there are any questions, please let me know.
Great. Well, thank you, Raluca. That was really very interesting. It's interesting to see all of the great projects that you're working on. I was just curious for the, maybe you could give us a sense of the timeline for the glucose oxidase project, for example. How long did it take you from kind of start to finish for that kind of a project?
So that project lasted 4 years. But at that time, we didn't have any experience either with the flow cytometry assays for enzymatic reactions or microfluidic devices. So I think the timeline should be smaller now, and we hope to get it accomplished in like 2 years time frame, a similar project.
Mm-hmm. And so there's certainly a lot of considerations that that you need to take into consideration for designing a project. How is it that people should kind of start if they're interested in doing this?
Well, I think, the easiest thing is to start a discussion with me, or if they already have some publications that are available, I mean, in general, if you are interested in the directed evolution approach or modifying an enzyme or a protein, the easiest thing to do is to find an assay that basically reflects that function. And once we have that, we can go from there. If there is no assay available or you're not sure about it, I mean, you can always come and talk to me. That's for sure. And even if there is something published, I mean, the likelihood is that it would need some optimization anyway. So.
Great. So there's a couple of questions in the chat. I'll read them to you, but if others want to add questions, or if you'd like to ask your own, you can just turn on your video or raise your hand and we can call on you. So the first question is, do you provide protein production service for academic use, for example, Cas9 protein?
We didn't specifically do Cas9. I mean, it is a bit tricky the way we are working at the moment. I do not have enough support staff, and the technicians and postdocs that I have hired are specifically hired for the industrial projects that we are working on, which means that the costs need to be supported by somewhere. We could help, and that's how I was working with other PIs where some of their postdocs or students were coming and working with me on specific projects and doing their work themselves on our instruments under my supervision. So that could be one model that we could work on. We don't necessarily provide, like a finalized product unless we have a project that is running for multiple years so that we can hire specialized people doing that and only that.
Next question is, do you provide phage display peptide screening?
We didn't do it so far. We were having it in the pipeline. However, we didn't need it until now. We were focusing on yeast surface display or E. coli libraries. We didn't do, or full antibodies actually, not on phage. We could do that, but as I said, we didn't really have a project that needed that specific application.
Okay, great. The next question is, does your group only identify and purify antibodies, or can you also manufacture novel antibodies?
Well, we can do screening for new antibodies, and I didn't present that. I mean, that's more of a scientific project that we are partially working on. We can do screening directly of hybridoma cell lines using microfluidic devices and actually identifying antibodies that are binding to a specific target. So that would be discovery of new antibodies in a high throughput screening method compared to the classical ELISA methods that most of the people are using. And then we could produce the antibodies at a higher scale. So at the moment, I think we are producing around 200 milligrams of antibody in a batch. I'm not sure if that answered your question.
It appears to have answered her question. So that's all the questions we have in the chat. If there's anyone that would like to ask a question, just either maybe show your face and unmute yourself and go ahead and ask. Maybe, Raluca, while we're waiting, you can post your contact information in the chat so people can find you, or your email address or your website address for your website.
Yes.
Can I ask a question?
Yes. Go ahead.
Yeah, so when you mutate that protein with error-prone, probably PCR, I guess, do you control how many mutations per gene, or it is just purely random?
No, we normally, when we start, we do a couple of reactions so that we do control the amount of mutations that we put per gene. But the amount of mutations per gene is dependent on each protein. So some protein can tolerate higher load of mutation versus some cannot. So it is again protein dependent. In most common cases, we have around 3 to 5 amino acid mutations per gene.
Okay, great. Thank you.
Welcome.
See, there any other questions? So it appears that you have answered all of the questions, and thank you very much for participating in our seminar series. I think this was a really great presentation, and I hope you get some future collaborations out of it. So thank you everyone for attending.
I posted my email, and if everybody has additional questions, we can always talk or shoot me an email, and I'm more than happy to discuss further. Thank you for inviting me today.
All right, thank you everyone.